Epigenetics is the study of biological mechanisms that switch genes on and off without altering the underlying DNA sequence. These changes can profoundly affect how cells read genes, subsequently impacting our development, physiology, and health. Epigenetic modifications can be influenced by a multitude of factors, including age, environment, lifestyle, and disease state. At its core, the concept of epigenetic changes revolves around how specific chemical compounds can attach to our DNA or to the proteins called histones around which DNA is wrapped. These compounds, or epigenetic marks, include methyl groups in a process known as DNA methylation and various modifications to histones, such as acetylation. These chemical changes affect the access of the cellular machinery to the underlying DNA, thereby controlling the process of gene expression. Epigenetic changes are a natural part of the human life cycle and play critical roles during development. As an embryo develops, epigenetic modifications help cells differentiate into the multitude of cell types that make up a human body, from brain cells to skin cells. However, epigenetic changes can also be a response to environmental factors. For instance, diet, stress, smoking, and exposure to pollutants can lead to alterations in our epigenetics. These modifications might not be immediately apparent, but over time, they can significantly influence an individual's risk of developing various health conditions. For example, certain patterns of DNA methylation have been associated with the risk of developing cancers. Tumor cells often exhibit significantly altered DNA methylation patterns compared to normal cells, which can result in the silencing of tumor suppressor genes or the activation of genes that promote cell growth. Research also indicates that epigenetic changes can contribute to genetic disorders such as Fragile X syndrome and various forms of mental retardation. In addition to cancer and genetic disorders, epigenetic changes have been linked to cardiovascular diseases, autoimmune diseases, neurodegenerative disorders, and mental health conditions like depression and schizophrenia. Furthermore, because epigenetic marks can be altered by lifestyle choices, there is considerable interest in how intentional changes in environment or behavior can potentially reverse harmful epigenetic changes and improve health outcomes. For example, epigenetic changes resulting from diet and obesity are a significant area of research, as they can affect the expression of genes involved in fat storage and metabolism. Studies show that a better diet and increased physical activity can modulate these epigenetic markers, potentially leading to improved health and reduced risk of metabolic disorders like type 2 diabetes. In another branch of health, prenatal and early life environmental factors, such as maternal nutrition, exposure to toxins, and stress levels, can bring about epigenetic changes that might influence the risk of developing diseases later in life. This underpins the concept of developmental programming, suggesting early life exposure can have long-lasting effects on an individual's epigenetic profile and disease susceptibility. The dynamic and reversible nature of epigenetic changes also opens up possibilities for targeted therapies that can reprogram the epigenome. Drugs that can reverse harmful epigenetic modifications are being investigated as potential treatments for cancer and other diseases. In conclusion, epigenetic changes, while a natural part of our biology, can significantly contribute to various health conditions, influenced by both our genetics and a wide range of external factors. Understanding and intervening in these changes hold the promise of new avenues for therapy, prevention, and personalized medicine, with implications across a vast spectrum of diseases and conditions.